Okay, well, now we are into chapter 10, and we are continuing on the topic of the senses and sensory transduction being the key word here. I got that coming up in a couple of slides here. And so in the previous chapter, we covered vision, and we saw there's quite a bit of research, both at the perceptual level, which really we haven't focused much on, but also at the neuroscience level of understanding vision. Out of all of the sensory systems, vision is by far the most heavily studied, and either touch or hearing comes in second. I would say historically, that is in psychology over the last 150-ish years, hearing was probably clearly the second most studied of the senses. But I think in recent years, there has been a very large kind of momentum shift towards interest in studying touch, but that's coming up later. Let's talk first about hearing. And as we did with vision, we kind of need to talk a little bit about the stimulus for vision, something about light, but then we're going to move into the details of how the sensory anatomy itself works, in this case, the ear, and then how that goes on back to the brain. So for hearing, we saw that previously, I should say for vision, photoreceptors in the eye are neurons, right? The rods and cones, but they are not typical neurons because they don't really have dendrites in the way we have seen them in sort of a standard case, but rather they have a specialized anatomical feature, these discs full of opsins, right? Chemicals that undergo a breakdown in the presence of light and changing the internal voltage of the cell, which then triggers graded potentials to the bipolar cells and so on and so forth. But in this case, we have a different kind of sensory neuron, a sensory neuron that has to respond to a stimulus now that is best described. Here I have called it a wave, but we may also think of it as a vibration because waves tend to be somewhat repetitive. We, of course, can hear some sounds that are very short and discrete, and therefore it's just maybe a single wave front that comes into the ear. But more often, especially if we're thinking about not just continuous sounds in our environment, but maybe speech and music, which are an important and common part of auditory perception, we're talking about patterns of sound waves that change over time. And that's very important when it comes to understanding how hearing works, is to understand how do the sound waves change over time and how does that carry information about a sound producing event so that we can recognize music and melody, we can recognize the human voice, we can recognize any kind of sound producing event. Now, a lot of this higher level perceptual stuff are things that we cannot answer at the neuroscientific level. And we saw similarly with vision that there's a lot of provocative findings with simple cells and things of that nature with vision. But beyond that, accounting for higher level conscious visual perception of objects is something that forces us to transition from neuroscience and physiological levels of explanation into perceptual theory and psychological levels of explanation. And I think the same will hold true here when we talk about hearing and really all of the senses once we get to those. But first, what is a sound wave here? Well, in this case, we just have a simple sound producing event. Imagine now striking a tuning fork and it plays a continuous note, right? So it's just going to ring out with a ding. And as what happens here is that when you strike the tuning fork, it compresses the forks together. And that compression of the two forks takes the air molecules that are floating around inside and outside of these forks. And between the forks, those air molecules become compressed. But then what happens is that as the forks then spread apart, the air molecules between the forks is now correspondingly spread out. And that term is called rarefication. So as the forks vibrate, those forks are vibrating. If you see my hands there, they're going in, out, in, out, in, out, in, out at some specific rate, depending on the size and thickness of the tuning forks, right? So you have different tuning forks for producing different musical notes, and some that are thinner will produce a higher tone, and others that are thicker will produce a lower tone, because that relates to the frequency of their vibration. And so a slower frequency of vibration, and that means here, how many times per second do these forks go in and out? So a slow frequency would be something like this, where I'm going really slowly in and out, right? But a higher frequency is when we go faster, right? And that results in a pressure wave now that travels through the air, because that first compression of the molecules is pushed out from the forks. And now that increased compressed dense air is now moving away from the tuning fork. And then as the forks pull back and rarefy the air between them, and then they come back again, that recompresses the air and creates a second compressed pattern of air that then also moves away from the tuning forks. And so we have, I used this term a second ago, wave fronts, right? The idea is that each compression pushes that dense air away from the forks. And this is happening now in a way that is very repetitive and also at an exactly consistent timing, right? And that's because tuning forks are designed to oscillate. That's what they're doing, oscillating at a very specific rate. And that corresponds to the frequency of the note that's being produced, right? So if it's something like a concert A, 440 or 444 hertz, whatever that is, right? 440 hertz, that means that these forks are vibrating 440 times per second. And these wave fronts that we see here on the right, these little curves that are moving out, are moving away from the forks at exactly that rate, 440 times per second. You will see these peaks of a wave moving away from the fork. Now, one way to help visualize these waves that are produced by this vibration, these kinds of waves, as they travel through the air, are at a physical level pretty much identical to the kind of wave that you would see on the surface of a pool of water. So imagine you just have a small pool of water, just like think of a little kiddie pool, right? A few feet across. And you were to drop a small stone into the middle of that pool. What would happen is that as the stone comes down and strikes the surface of the water, that compresses the water, right? And that pushes the water up in a circle around that point. And that circular compression pushes, as it pushes the water up, causes a peak, right? And that peak of a wavefront now travels out in a circle in all directions from the point at which you had dropped the stone, right? And right behind that peak is actually a small trough, a dip, because that's the rarefication that occurs right behind it, right? And the same thing is happening here, is that if you just created a single sound, like if I clap my hands, we'd get that same stone dropping into the pool. We would have a single peak followed by a rarefication right behind it of air molecules now traveling out, not in a circle like on the surface of a pool, but now in a sphere, because of that it travels in all directions away from the point where the compression occurs, right? But the pool serves as a good analogy because we can try to visualize the wave. Imagine if you could, and obviously we can't necessarily, put your eye right at the level of the surface of the water, and you could see this wave traveling across the surface, and you would see the peak, and tracing my hand up, right, followed by the trough, right, and we get that kind of wave pattern, right? And now in air, we cannot see that wave, right, because it's just a compression of the molecules, and therefore we could measure it with something that measures air pressure, right? Like a barometer can measure air pressure, right? And that's what we're really seeing here as waves of pressure waves that are traveling through the air here, compressing and rarefying all of the various molecules, oxygen, nitrogen, carbon dioxide, all of that stuff, dust and moisture, all of the stuff that's found in the air around us, right? Here's that waveform that I'm talking about. So again, we can visualize the wave in this way as a series of peaks and valleys. So in the case, like I said, of just clapping my hands or dropping a single stone in the pool, you're going to get one single peak followed by the trough. But anything that's repetitively vibrating, like the tuning fork, is going to cause this continuous vibration and this continuous series of peaks traveling away from the tuning forks in all directions, right? Likewise, if I were to, I guess, repeatedly drop little pebbles into the pool at some specific rate, like 100 times per second, I'm dropping small little pebbles into the top of the pool here, then you would see series of wave circles traveling out 100 times per second, right? So what this graph represents here is on the y-axis is air pressure, right? So again, it's pressure compressing the air. We increase on the y-axis there towards that positive increase. And then, of course, the rarefications would be the low pressure component of the waveform that follows right behind it, right? Time is on the x-axis here. So what we have here is this idea of being able to measure how many peaks are occurring in a specific amount of time. And that's important because I'm using this concept I said of frequency. So the idea is you could have a tuning fork that rings out at 440 hertz. And what that really means is that it's ringing at 440 times per second, right? And that means there would be 440 of these peaks in the wave every second. Now in this graph, on the other hand, that's clearly not a 440 hertz tone. How many peaks are occurring in 1 second in this graph? Well, it looks like pretty much just one, or maybe even not an entire one, right? Only a partial peak occurs within the first second here, right? It looks like a full cycle. A full cycle is usually the idea of starting in one location of that curve and then going to the point where it repeats itself. So if you look on the top here, what you see is that the graph starts at a trough, right? So when x is equal to 0, at time equals 0, we're at a trough. It travels up to the peak and then it drops back down to a trough. And then as it starts its way back up again, we're repeating the cycle, right? So in that way, the graph, you could kind of look at that graph as trough to trough. And that's the way they kind of chop that out of there. So in the picture in the middle, they just took one peak, and see on the left and on the right, that's where it kind of goes back to where it started, right? And that's one full cycle, right? So from peak to trough to back to, sorry, I should say from trough to peak and back to trough is one full cycle, right? And that in that case, to understand the frequency of a tone, we would want to know how many of those happen in 1 second. In this case, I would kind of guesstimate from looking at the graph that it's about 3/4 of a cycle. And so in this case, we would say this has a frequency of 0.75 hertz, right? Just 3/4 of a single cycle happened within 1 second. That's not too crucial for our immediate purposes here, but just to give you an idea of what the frequency is referring to here. And so in fact, there are maybe 3 components of a sound, physical components of a sound wave, that we want to talk about. And that is the first one right here is frequency. How many cycles repeat themselves in some specific period of time? And of course, most likely we're interested in how many cycles occur within a given second. And that's officially measured using the unit hertz, right? Abbreviated Hz. So as I said, a 440 hertz tone is 440 peaks per second. A 1,000 hertz tone is 1,000 times per second. 10,000 hertz tone is 10,000 times per second, right? The human ear, I'll mention this now because I don't have it anywhere else in the notes here, can only hear a certain range of frequencies, right? So there are some very, very low frequencies that the ear cannot simply detect. And that would be anything below 20 hertz, right? So a 10 hertz tone, some kind of a sound that only creates a vibration 10 times per second simply is not detected by the human ear. Even a 20 hertz tone, which is the bottom end, is very difficult to detect. And it really just sounds like a very low, deep rumble, right? It doesn't really sound like anything like a musical note or anything like that. Meanwhile, the upper range of human hearing tops out at around 20,000 hertz, but that's really only the case in a young person who has no particular problems with hearing. Once we get older, we start to lose that upper end a little bit, and we kind of drops down to maybe 17,000 or 15,000 hertz as the upper limit of your hearing, which in most cases is not particularly problematic because there are very few sounds in our environment that really hit those higher frequencies. And certainly most of the things that we might be more interested in hearing, like say music and the human voice, don't get anywhere near that, that kind of stuff, right? Now the amplitude. So the amplitude was on that, was the y-axis on that graph of the waveform, right? How high is the peak? So again, let's go back to the pool metaphor. If I drop a small little pebble into our pool, it's going to make a tiny little splash, right? And you're just going to get this tiny little wave that travels out in a circle. But if I take a big rock and I drop that into the pool, then clearly I get a bigger splash, bigger wave travels away from it, right? And that's the amplitude. How tall is the wave is essentially, and that's based on how much force and pressure was applied at the point of contact with that rock hitting the surface of the water. And the same thing happens with sound, is that if I strike the tuning fork very, very lightly, I'm still going to get the same frequency, but now they vibrate at a small amplitude. They don't move very far. They move at the same rate, but they don't move very far. But if I really whack it really hard, then those things compress really hard together. You get this hard compression, and then you get a higher amplitude wave. And so, of course, clapping my hands, it's the difference between a light clap and a really heavy clap, right? Once I put more force into it, I'm compressing to get higher amplitudes, and that relates to the height of the wave, right? Now we can measure the amplitude physically as well using certain measurements. As I said, you could use something like a barometer that measures the what's called the sound pressure level, right? And so the pressure level would be the actual compression and barometric pressure of this wave. But one of the tricks here is that we don't frequently use that kind of unit, like what we call SPL units, sound pressure level units, because they vary quite dramatically, right? They can be fairly small, but they can go up to sort of like an order of magnitude in terms of the range that we can detect. And so the decibel scale is easier to work with. So the decibel scale uses a logarithmic function to compress this range of SPLs that we can hear into a simple linear scale. And the way it works is by using a reference tone that is the absolute threshold, the weakest tone that we can hear. And what this does is it ensures that the value of 0 dB is essentially our absolute threshold, right? Anything at 0 dB is the weakest stimulus that we can detect. So clearly then that means that 0 dB does not mean the absence of sound. It means the weakest sound that we can hear, which actually implies that you can have sounds with negative decibel levels, right? Because there can be a sound that is present, but it is below our threshold, and then it would have negative dB values, right? But the idea is that every time we double the sound pressure level, we increase the decibel value by a factor of 6, right? And so 6 dB is technically twice as much amplitude of sound pressure as 0 dB, and then 12 dB is twice as much as that, and then 18 dB is twice as much as 12, right? It's a little bit counterintuitive, but that's the way that log function works here, right? The upper limit of human hearing is in the ballpark of 120 decibels. Once we get around 120, 130, we're so loud it's like standing under a jet engine. And it's the point where it would actually just damage your ears to the point of no return there, right? Now, one last thing to say about the physical properties of a sound wave is what we might call the complexity of the tone. And this relates to the difference between what we call a simple or a single tone, pure tone, or a complex tone, right? And in this case, pure tones are pretty rare. They don't not pretty much occur in nature, but with a tuning fork, as I had on the previous example, that comes close. It's not exactly a pure tone, but when I described it as maybe emitting a tone of 440 hertz, if it really was just that perfect of hitting exactly 440 hertz and it had no other frequency components as part of it, then it would in fact be what we'll call a pure tone, right? We can use other advanced forms of technology like an oscilloscope. So an oscilloscope, which are used in synthesizers and things like that, can generate pure tones, right? So you can get any pure tone you want. If you want a 1,000 hertz tone, a 1,500 hertz tone, then you can generate those, right? And we can do it with computers too, I guess, right? That's my old-fashioned approach there, thinking about analog oscilloscopes and synthesizers, but still you could do it on a computer. In fact, if you want to play around, you can go onto YouTube, and for whatever reason, there are YouTube channels that have examples of pure tones of different frequencies, and you can just listen to them. And sometimes in a classroom environment, I'll do that, but we're not going to do that in this recorded lecture. But as I said, in real life, the sound of someone playing a musical instrument is not just a pure tone, right? You don't, because a pure tones just sound like a dee, right? It doesn't have any particular complexity and quality to it. It's just a single note. And most sounds in our environments are actually a layering of many different pure tones, all blended together to create what we call now the complex tone. And this is, for example, what distinguishes the difference between, say, a brass instrument like a trumpet or a trombone from a woodwind instrument like a clarinet or a saxophone, is that you get, you maybe might have a certain fundamental tone. So the fundamental would be the kind of like the bass note of the note that's being played, and that determines maybe the pitch and the general quality of the musical note. But there's what are called overtones, right? That there's other higher frequencies that are present oftentimes in certain intervals or ratios. And the nature of a brass instrument produces different overtones than the nature of a woodwind instrument. And that gives us the unique quality, what's sometimes referred to as the timbre of the instrument, right? And so a saxophone is easily distinguishable from a trumpet because of that timbre, which relates to the nature of the overtones that are present. And the same, of course, applies to the human voice, right? That when our vocal cords vibrate, it's not producing just a pure tone, but it's producing many different tones that resonate through the oral cavity. And then when we sing, of course, you can do things. If you take vocal training, you can learn about singing in the head voice, where you're trying to get more resonance up in the head. You can have a nasal sound where you're getting more resonance up in the sinus cavity, or you can sing from your throat, or people sometimes call a chest singer because of the way they can kind of belt from their chest. But, you know, all these details of musical performance, we don't necessarily need to worry about. But the idea is you can get different qualities of the sound based on not just the fundamental frequency that's present, but also the nature of the overtones and how that changes our perception. But the reason I'm talking about this, though, is that turns out to be pretty important for recognition, right? At a perceptual level, how do I distinguish a trumpet from a voice from a clarinet? Well, it's because the ear seems to have some particular ability to distinguish those overtone patterns, right? Here are some examples that show us what that looks like in graphical form here. So on the top, we can see the difference between a low frequency and a high frequency tone. So when we make a graph of these waves, low frequencies show the waves as being more spaced out and farther apart. High frequencies show those waveforms as being compressed and all close up together, right? When it comes to amplitude, we got a high amplitude with the peaks being highly spaced, and a low amplitude with the peaks are not spaced, sorry, but just larger, taller, right? And low amplitude would be the peaks being a little bit smaller there, right? Notice the spacing is the same. The spacing between the peaks is the same, so that what's different there in the middle is just the difference in the amplitude of those waves. Now, at the bottom shows an issue of complexity, right? So a simple tone is really just a single wave. We can use a trigonometric function like a sine wave to describe these. So we can also sometimes call this a sine wave tone, a pure sine wave. But what happens with a complex tone is that we might take several of these pure tones, sine wave frequencies, and we're just going to blend them all together. And of course, it would look like that if I just superimpose them all together. But on the other hand, if you just were recording this tone and tried to make a graph, it would just result in a very messy looking graph with lots of peaks and valleys, kind of at many frequencies and many amplitudes, all just kind of messed up with each other. And that is an important part of, as I said, higher level auditory perception. So getting into the transduction part, as we said, so previously with vision, we had to have specialized neurons that convert the electromagnetic energy, the lights, into a nerve impulse. And now what we have to do is we have to convert these pressure waves into a nerve impulse. And what we have to do is we first have to transmit the sound, pressure, vibration in the air through the moving parts of the ear to activate those special neurons that we have that will respond to these waveforms somewhere in the inner ear, right? So we can describe the vibration here as mechanical energy if you want to, because it is, of course, a movement of air molecules, right? And then we're going to process that information so that we can start to perceive things. So as I said before, we can do a lot of things with hearing. We can identify what it is. You know, is it a musical instrument? Is it a human voice? Is it a car engine? Is it a bird singing? You know, what is it? We can also localize sound sources. So that's important. We want to understand. And actually, even though I was speaking somewhat pessimistically earlier about our ability to neuroscientifically explain higher level perceptual processes, we have some fairly good neuroscientific explanations about how we achieve sound localization. So that's interesting. And then also, though, of course, it's one thing to simply recognize that a sound is a human voice, but it's an entire other thing to decode the complex pattern of sounds when a person is speaking so that you can recognize the words that are present. One of the reasons why this is challenging is because people have different voices that vary in their frequency, that vary in their speaking rate, that vary in their intonation pattern, that vary in their accent and way in which they pronounce certain sounds. And yet we can get past all of that variation and variability in human speech to still get at the underlying, those are the sounds and words that someone is saying. And ultimately, of course, is to get to the meaning of the language. And to add more psychological complexity to that, getting past the messiness of how different people speak differently, we also do a lot of other weird things with language, like we speak in metaphors sometimes. And that means that the literal interpretation of what I'm saying is not what I'm intending to say. And I'm saying something else through metaphor or sometimes through sarcasm and so on and so forth. And so there's a lot of higher level complex perceptual and cognitive processes that have to be involved to truly decode language, right? And we really can't tackle all of that. That's a whole big thing, right? Not in this one chapter anyway. And then similar to that is recognizing a song, right? So it's one thing to recognize that I'm hearing horns or, whether it's brass or woodwind, or that I'm hearing drums or hearing guitar or piano or whatever it might be. But I can recognize melodies and rhythms, and therefore I can recognize a song, and it doesn't matter what song it is, right? Or sorry, I'm not trying to say it that way. What instruments it's played on, right? Like you can play a familiar song on piano and you can recognize that song. I can switch to playing it on guitar and you can still recognize the song, even though the overtones are different. And sometimes you can even change the key, right? So you can transpose the key to a different key. But you can still recognize it. And that's because oftentimes it's not about perceiving the absolute notes that are present, but the intervals between the notes are really the more important part for recognizing certain melodies. And the same could probably be said about rhythms. Sometimes it's not about the specific beats, but it's about the intervals between the beats. And this kind of pattern recognition is something that requires a pattern that unfolds over time, and our ability to track that over time is, again, a complex, higher level perceptual process. Let's get into the anatomy of the ear, though. So first thing we're going to do is subdivide the ear into the outer, middle, and inner parts, and that's fairly simple enough. And then we have a list of anatomical structures. So the outer ear consists of the pinna, the auditory canal or external ear canal, and the eardrum. And the eardrum can be seen sort of as a boundary between the outer ear and middle ear, but officially we'll list it under the outer ear. The middle ear will just be pretty simple. It's the three bones that we know as the hammer, anvil, and stirrup, or we can use their Latin names. Malleus, incus, and stapes, right? But doesn't matter. Now these connect up to the oval window of the cochlea. So the cochlea now is the inner ear, and it has listed here its parts. So we have just a list, and I'm not going to read through the list here, but what I want to focus on here is that there are these hair cells inside of the cochlea, and these are those special neurons, right? So neurons that are called hair cells have, in place of dendrites, small little hair appendages that stick out from the cell body. And anything that moves that hair, I'll use my finger here, right? If it moves this way, or sometimes something might come along and press it down and compress it, right? That is a mechanical deformation of the hair, and that triggers an action potential in these auditory neurons. What we want to understand here is there's a couple of membranes, right? And one of these membranes listed there is the basilar membrane, and another one, tectorial membrane. And so the basilar membrane and the tectorial membrane, I'm going to use my hands here as examples of that, kind of lay very close to each other, right? And the hairs of the hair cells kind of protrude out into that space between those two membranes. And as this vibratory wave travels through the outer ear, through the pinna ear canal, causes the eardrum to vibrate, and then that vibration is transmitted through the three bones into the cochlea. And this cochlea is this fluid-filled organ with these membranes inside of it. And as these membranes vibrate like this, the little hair cells in between them are getting squashed, right? And that's what's going to trigger the action potential in the neurons. And I've got some pictures of all of that right here. So we see again, we have that tuning fork emitting its repeating wave fronts as they travel. The pinna, think of the pinna as essentially like a sound funnel, right? It's got all these folds and they're pointing in many different directions, which helps take sound from different directions and funnel them down into the ear canal. Do we want to use an analogy of the cornea and lens of the eye? I don't know, that's possibly a stretch, but something like that. I guess the goal is to get that sound into the ear, right? So then the wave travels through the ear canal. It causes the eardrum to vibrate. Behind the eardrum, we find the three bones listed here as the ossicles, right? And we see in the close-up view, what happens is that the base of the hammer bone is attached to the backside of the eardrum. And so as the eardrum is vibrating as a result of that sound wave, the hammer bone also vibrates, and then it strikes the anvil bone, which in turn vibrates, and then it pulls at the base of the stapes, or the stirrup bone, and that also vibrates. And that pulling of the stirrup bone on what's called the oval window, which is a pretty obvious anatomical feature, an oval-shaped hole into the base of the cochlea, causes that vibration to be transmitted into the cochlea, which, as I said, is a fluid-filled organ. It's shaped kind of like a snail shell here. And so we have these coils, these winding coils full of fluid. Now, inside the cochlea, as we go down to the bottom right here, we're going to see that the interior lining of the cochlea is really three different chambers. They're all filled with fluid, so that vibratory wave is going to travel through these fluid-filled chambers. But at the boundary between two of those chambers, we see those two membranes, right? So the bottom membrane here, kind of shown in purple there, is the basilar membrane, right? And we can see from the basilar membrane that it is embedded with these hair cells, right? And the hairs are sticking up out of the top of the basilar membrane. Meanwhile, above it in blue is the tectorial membrane. And the tectorial membrane is a little more rigid. So I said before that they're kind of both flapping in response to these waves, but really it's the tectorial membrane that sits there kind of rigidly, and the basilar membrane is the one that vibrates up into it, right? And that's what's really happening here. And so what we're going to do now is we can see that little magnified view of just a single hair cell that has these tiny little hairs that stick up. We'll call them cilia, right? But these little hairs that protrude out from the base of that neuron, those cilia are the things that are getting smashed as the basilar membrane vibrates and moves up and collides with the tectorial membrane, right? But it's this entire thing, the tectorial membrane combined with the basilar membrane that we call and all those hair cells is what we call the organ of Corti. This is really where the action is for hearing, right? Now, a couple of things we want to say here. So we can see that the hair cells have axons, and the axons you can kind of see as those yellow fibers on the bottom that are leaving the cochlea to form the auditory nerve. And in the top right, we see the auditory nerve then kind of traveling away from the cochlea. But I didn't say anything about those semicircular canals, right, that are right there, pretty obviously attached to the cochlea. So what are they, and do they have anything to do with hearing? And the answer is no. That is part of the vestibular system, which you may know as your sense of balance, which may be better referred to as an accelerometer. And we're not going to talk about that in any detail here, because that's for later when we want to talk about the body senses and touch. But it's worth pointing out that one of the reasons why it is attached to the cochlea is that the receptors inside the vestibular organs are also hair cells that are very similar to the hair cells for hearing. But in this case, these hair cells are not stimulated by sound waves. They are stimulated by the movement of fluids inside of these organs, which occurs when we move our bodies around and accelerate the bodies and turn in circles and things like that. That causes fluid to slosh around inside those canals and will stimulate those hairs, those cilia on those hair cells, right? But let's move on. Here's again the cross-sectional view of the cochlea so that we can get a good sense of those two membranes that are kind of lying up against each other, the tectorial membrane and the basilar membrane embedded with the hair cells. And again, as I said, the tectorial membrane is fairly rigid, and it's the basilar membrane that's going to do most of the vibrating up and down, and that then results in we'll call it shearing, technically, of the cilia, the bending and shearing of the cilia as a result of these sound wave vibrations that are directly influencing the basilar membrane. I think we got that down here. Now, next question is, based on how the inner ear and the cochlea and the basilar membrane and all these kind of stuff, how it responds, helps us understand perhaps how the ear is able to encode certain information about the pitch of a tone, what's the frequency, right? And so is it low pitch, high pitch, and also something about the loudness perhaps. But we're going to start first by talking about pitch perception, frequency and pitch perception. And this goes back to von Bekesi, although the idea had been proposed maybe sometime before then, but von Bekesi was one who really really figured this out in detail. And what von Bekesi realized is that different regions of the basilar membrane, so imagine the basilar membrane, let's go back to this picture real quick, it's layered within the cochlea, but the cochlea spirals around. So the basilar membrane itself, of course, is spiraling around in circles here through those chambers of the cochlea. And as it turns out, at one end of the basilar membrane, it has a different thickness than at the other end. Right, it gets thinner the further we go from one end to the other. Right, and as it turns out, one end of the basilar membrane responds to these high frequency tones, and it vibrates stronger. Right, it vibrates more. It's more sensitive to vibrate when it's exposed to high frequency tones, and then the opposite end correspondingly will not vibrate very much in response to these high frequency notes. Conversely, that opposite end vibrates more to a low frequency, right? So let's see that here. What we've done here is we have to imagine uncoiling the cochlea. So if we take that snail-shaped organ and we roll it out so that the basilar membrane was laying out flat instead of wrapped in coils inside of there, right? We see exactly what I just said, that at the base end where the oval window is, it says it's a narrow, thick base, right? And it thins as we get farther to the opposite end. The opposite end is called the helicotrema, but I guess we don't need to know that. But at the helicotrema end, it gets thinner, and that end vibrates more for low frequency. So that thick base is more responsive to high frequency notes. The thin far end of it is more responsive to low frequencies. And so then, of course, anything in between would be any of those sounds in between. So as I said earlier, 20 hertz is the lowest sound that we can hear. That has a lot to do with the response of the thinner end of the basilar membrane. It will vibrate to a 20 hertz tone, but it will not vibrate in response to a 10 hertz tone. It simply just doesn't react to that. You need a thinner membrane for that, right? Likewise, at the thicker end, that far thick end will in fact vibrate in response to 20 hertz tones, but it doesn't respond to anything higher than that, right? So we call this the place theory of pitch perception, because the idea here is that our perception of pitch is a function of which place along the length of the basilar membrane is now being stimulated by the sound, right? So you get sounds of different frequencies coming in, and you're going to get different places down the length of the membrane that are vibrating in response to those different frequencies. And in turn, that's also going to activate different populations of hair cells, right? Because it's a totally different population of hair cells down at the oval window end of the basilar membrane compared to the population of hair cells down at the helicotrema end of the basilar membrane. And then you're going to have differential neural responses as a result of the differential basilar membrane response. And that differential neural response gives us different perceived pitches, right? So let's come back to the previous slide, right? So that's the idea here, is that we're calling this the place theory of pitch perception, because which place and therefore which population of hair cells get stimulated varies down the length of the basilar membrane as a function of the frequency of the tone, which is then encoded as a pitch. Next thing we want to know is, where does the signal go as it leaves the ear? And so what's happening is that these hair cells send, kind of like with the rods and cones, they send graded potentials to the bipolar cells. Well, there are bipolar cells in the cochlea as well. And so these hair cells send graded potentials to the bipolar cells. We can get some degree of spatial and temporal summation occurring on those bipolar cells, but we're not going to try to map all that out. And then that forms, the axons of these bipolar cells now form the auditory nerve, and it goes to the brainstem, right? So we have something called the cochlear nucleus. Remember, a nucleus is a cluster of cells in the central nervous system. So it's receiving input in the brainstem and the medulla. But there are other connections that travel to other locations in the brainstem, including something called the superior olive or olivary complex, however you want to call it. And very close to that is something called the trapezoid body. And we're going to talk about what those do coming up soon, right? But a little later in a couple of slides down the line here. And then also to the midbrain, so the inferior colliculus in the midbrain. And one of the things that you might know about the inferior colliculus, I think we may have covered it before, or maybe we'll cover it later, is that it helps control some of what are called orienting responses, right? So if I make a loud sound and you kind of suddenly kind of jump and startle and you look in the direction of the loud sound, that's an orienting reflex, and it involves that part of the midbrain, the inferior colliculus, right? Obviously, it requires us to have to localize where the sound source is coming from, and that's actually what the superior olive and trapezoid body are doing, is it helps us determine the direction and location of a sound source. But then the inferior colliculus is going to help control like the head and neck muscles to get our head moving in that direction, right? But meanwhile, of course, it also further allows the nerve path to project on up to the brain, where it has to go to the thalamus. Remember, the thalamus redirects sensory input to appropriate areas of the cortex. And in this case, we have something called the MGN, or medial geniculate nucleus. Remember, we had a lateral geniculate nucleus for the visual pathways, and lateral means more off to the sides of the thalamus, and the medial geniculate nucleus would be more towards the middle of the thalamus, right? And what's also interesting here is that the MGN is actually divided into two parts, and one is ventral, that means it's just towards the bottom, right? And the other one is dorsal, which means it's more towards the top of the thalamus. And so we have a kind of a separate double pathway here, a ventral MGN signal and a dorsal MGN signal. What do these mean? Well, primarily the ventral signal goes to what's called A1, and that's the primary auditory cortex. Remember, with vision, we had V1, right? Primary visual cortex. So there's an A1 here. And that means it's the very first area of the brain to receive primary auditory processing information. It's in the temporal lobe of the brain. The dorsal MGM goes to other areas kind of near A1, but it also goes, and those would be in the temporal lobe, but it also goes into the parietal lobe of the brain, specifically the posterior parietal cortex. And this leads us to speculate, oh, just like vision had signals that went ventral into the temporal lobe and dorsal into the posterior parietal cortex. Now hearing does as well. Does that mean hearing could also be subdivided into the so-called what and where channels, right? So they have separate auditory pathways for recognizing what the sound is versus where it is. Yes, that's the idea, exactly what the idea is. Though there's a lot less research on understanding the what-where pathways for hearing than there is for vision. Next up, we want to talk about the organization of the auditory cortex. So we have A1, the primary auditory cortex, and the temporal lobe and a gyrus called Heschel's gyrus. There's also a secondary auditory cortex, which kind of surrounds and spans around that area. We can also call it the planum temporale. It has a lot of areas that are part of what it does, but one of the parts of the secondary auditory cortex is Wernicke's area, right? And so Wernicke's area, we've mentioned briefly in the past, is part of speech perception and comprehension, right? Our knowledge of the meanings of words frequently. And so, of course, as I said, when we hear language, it's one thing to recognize that I'm hearing a human voice, but it's another thing entirely to decode that, to know the sounds and the words and recognize the meaning and comprehend the language. And that particularly involves Wernicke's area. As we've learned before, people who have damage to Wernicke's area have great difficulty producing or understanding, speech. It also would make some sense, though, that Wernicke receives input from the occipital lobe as well, because, of course, when it comes to reading the written word, called the orthographic input, that would need to access the same information that as far in terms of listening to language or reading language, right? Of course, after we comprehend the meaning of either text or the spoken word, we might want to respond to that in some way. So, of course, there would be a pathway that takes us from Wernicke's area to Broca's area in the frontal lobe, so that that's where we might find all of the speech production and grammar side of stuff, which is very close to the motor areas. There's an equivalent region of A2 on the right hemisphere, but it's not typically involved in language. So in the left hemisphere, for most people, these are the language side. So that left A2 is going to give us Wernicke's area, and the left frontal lobe is going to contain Broca's area. But on the right side of the brain, those equivalent regions tend to be more focused on music perception and production, right? So a couple examples here. For example, we have the study from the 90s where they did PET scans, right? And what they find is that if you just present noise, this is just, like white noise, pink noise, just staticky sounds and things like that, it activates primary auditory cortex, A1, right? But if I play recordings of people speaking words, then we get activation of A2 in Wernicke's area in particular, right? If I ask people to not just listen, but I want them to actually engage in some sort of active task that involves discriminating different kinds of sounds, like can you tell the difference between a ba, ga kind of sound or something like that? We also see activation in Broca's area, and that seems to suggest that in part recognizing those subtle differences in sound might involve what we call a motor theory of speech perception, right? The idea is that I recognize the difference between ba and ga because I understand the kind of movements that I would have to make in order to make those sounds myself. And so I need to access Broca's area in order to distinguish that. Conversely, of course, if I just play musical stuff, melodies, we get A2 on the right side of the brain. So as I said, there's musical processing on the right side of the brain. And curiously, if I change it to a more of an active listening task where now I want people to not just listen passively to music, but to actually discriminate different musical notes. So I'm going to give you a task where you have to say which musical note is higher or lower than a standard tone or something like that. Suddenly, we get the right frontal lobe in a region that is very similar to Broca's area in its location, but now on the right side of the brain instead of the left. I should add, by the way, and I did have this little brief mention on the previous slide, so I'm going to jump back. This idea that language is on the left side of the brain and some of this musical stuff is on the right side of the brain is mostly true just for right-handed people. But it's not 100% true. And it's also kind of true sometimes for left-handed people, but there's some more exceptions. So this laterality distinction in the brain is more of a good rule of thumb, but not 100%. Here's a little flow chart that'll show us some of the things we're talking about. So on the left here, A1 is shown in the slight purplish shading there, small region, right? And then A2 is that larger yellow area there, including that one place where we're pointing is Wernicke's area, and that's for speech comprehension. What we're also seeing on the right is a pathway that's just represented by the arrow that sends information from Wernicke's area to Brokaw's area in the frontal lobe for the speech production kind of stuff. And it connects up. So in the motor area, shown in that light blue, down at the bottom lower regions of the motor area, those are the neurons that are important for controlling the speech articulators, the lips, tongue, and jaw, and things of that nature. So the pathway that connects the Wernicke to Brokaw's area is called the arcuate fasciculus. And so these three things together kind of really represent the overall speech part of the brain, between Wernicke's area, the arcuate fasciculus, and Brokaw's area, as well as the motor parts there in motor cortex. That's a lot of the speech stuff. So of course, this does leave out the reading side of things, though. One thing else we want to say about A1 is it contains what we call now a tautotopic map. So when we talked previously a little bit about motor areas and somatosensory areas and just mapping out a little bit of the brain, we said that these map out the body. And as it turns out, primary auditory cortex maps out pitches, right? So there's regions of, as we have to think back to the basilar membrane, right? So the idea is that one end of the basilar membrane responds to these high frequency tones and the other end responds to the low frequency tones, and that activates different populations of the hair cells. Well, those distinct populations and of hair cells that are stimulated by those different frequencies are preserved in the signal from the auditory nerve up through the MGN and up into the A1, right? And that means different regions of A1 are receiving activation as a result of a low pitch versus a high pitch. And so we can see differential activation of neurons in A1 that correspond to these different frequencies and different pitches of tones, right? Here is a picture that kind of shows that. The way that this gyrus works is that it helps to visualize the gyrus by doing a little bit of retraction of the frontal lobe so that we can kind of see into that sulcus there to see the primary auditory cortex. And here we see how it's mapped out with the left side corresponding to the lower frequency tones that we can hear. Here it calls it the apex of the cochlea. I previously called that the helicotrema, but it's the farther thin end of the basilar membrane. And the on the right with those higher frequencies, that's the oval window end of the basilar membrane. Now, this idea of place theory and how it activates these populations of neurons that is then preserved all the way back to the A1 organization helps us understand some things about how pitches are perceived, low and high and medium pitch sounds. But how does the ear encode the loudness of the sound? That's a different issue, right? So it's one thing for a tone to be low pitch, medium, or high pitch in nature, but we can also distinguish quiet versus loud or medium sounds, and that has to somehow be encoded in this nerve signal and processed by the brain. So we might ask, how do we achieve that? So we have a greater, now with loudness, we have that higher amplitude wave coming in. One of the arguments here is that what happens is that these cells, the hair cells and the bipolar cells in the cochlea, instead of, now it's not about which cells are active, which would be encoding pitch information, but it's now about a faster firing rate. So as the sound gets louder, these neurons fire faster, right? And now that is encoded as a signal that's interpreted by the brain as saying, okay, faster firing means louder sound, right? And of course, that what that would also entail then is that it's not just the bipolar cells that are firing faster, but when we look at the neurons in A1, they are also firing faster, right? Now, here is a mapping of all of this kind of stuff, and we can see how this works, right? So cross-lateralization is something we want to see here. So we see the signal coming in from the left ear is encoded in red, and it travels to the brain stem, and we see the activation of the cochlear nucleus, right? It's part of the medulla, and from there it travels up to the superior olive, and then to the inferior colliculus, and then up to the MGN, medial geniculate nucleus, and then finally up to the auditory cortex. But we see that there's a crossing over in a way that looks very much like what happens with vision, right? So that information from the left ear is traveling both to the left temporal lobe and crossing over to the right temporal lobe, right? And this occurs as we are traveling between the brainstem and the midbrain and the inferior colliculus, right? And so likewise, the right ear is encoded in that blue arrow, and that again is giving information that crosses over at the inferior colliculus so that it goes to both sides of the brain. Now, notice that even though the crossover occurs as it travels up to the brain, it occurs between the superior olive and the inferior colliculus. There's also a separate crossover that's happening down at the lower levels between the cochlear nucleus and the superior olive. But that crossing over has a different function entirely. And so, as I said earlier, the superior olive, one of its main jobs is to help us localize sounds. So let's understand how we localize sounds. What's really happening here is that when a sound is to your left, let's just say, that sound wave arrives at the left ear a fraction of a second before it arrives at the right ear. And this means that there's what's called an interaural arrival time difference, right? And the ability to detect the arrival time of a sound to the left ear versus the right ear requires that these two sounds have to be compared somewhere in the brain. And that's what's happening with the superior olive here, is that each superior olive is receiving input from the cochlear nucleus from both ears, right? And therefore it's comparing the timing of these two inputs. And if the superior olive detects that the signal comes from the left ear just a little bit before the right ear, then it knows that, oh, that sound arrived over from the left side of the head, right? Or conversely, if it detects that the sound has arrived to the right ear first, just a fraction of a second before the left ear, then again, the answer is that we know it comes from the right side of the head. There's also a loudness comparison as well. So the superior olive and the trapezoidal body are also comparing the intensity differences, because a sound on your left is going to be louder in the left ear compared to the right ear. And so again, by comparing these intensity differences, the superior olive and the trapezoidal body will be able to make that comparison and say, okay, we're confirming, yes, that's definitely over here on the left or definitely on the right. Which is then why, once that determination has been made, the signal travels up to the inferior colliculus, because if I want to turn my head, especially if it's a loud sound suddenly occurring and I need to orient myself to say, what in the world was that? And I look to the left, then the inferior colliculus is going to play a role in a kind of a reflexive response, that orienting reflex that we have, right? So that's an important part of being able to localize sound, is by comparing these arrival time differences and intensity differences between the two ears, which happens at this very low level kind of brainstem comparison there, right? All right. And lastly, this is actually somewhat repetitive, redundant with what I said earlier, right? But there's this idea that, that if we really are separating a dorsal and ventral MGM channel, that is used separately for recognizing what a sound source is versus where it is, then of course that is something that is we're going to comment on here. But as I said earlier, there's so much research on vision, and there's a lot of research on looking at how that works in visual research. There's quite a bit less when it comes to understanding the auditory response. So it becomes a lot more of a really kind of a hypothesis at this point, that auditory information really is kind of streamed in this functionally different way. But it's worth having a little note of here, and that pretty much covers this chapter. So we will call it, and that's it.